Are you tired of cramped hands from pipetting for limiting dilution? Of booking hours on the fax machine to sort single cells? Of hunching over a microscope, miserably squinting for clones? Of your cells dying after isolation? Are you unhappy with spending hours in the lab and thousands of dollars only to meet with failure and not get the clone you want? We and our cells have suffered enough. We need a better highway for developing cell lines than subjecting ourselves to the torture of limiting dilution and sorting. Current methods for isolating single cells and growing them into colonies lead to fragile and dead cells, resulting in poor outcomes and a long, bumpy road to desirable clones that's paved with expensive specialized instrumentation, miles of plasticware and microwell plates, hours of lost productivity, and liters of specialized media. Take the exit ramp and make your scientific journey smoother with CellRaft technology. Using this unique CellRaft technology, we've developed cell lines from some of the hardest cell lines out there, including suspension cells, primary cells, induced pluripotent stem cells, and even 3D organoids. So what makes this technology different? The number one issue that cells face in all of these methods is a lonely solo road trip to a microwell and a huge barrier to overcome in growth and viability. In our consumable, the CellRaft array, your cells are single but never alone, because they're cultured in a flask-like culture environment that also provides for spatial segregation of the cells within individual microwells. The cells are attached to a microscale growth surface called the CellRaft, which allows for crosstalk between the cells, leading to a high cell viability and superior clonal outgrowth. Within a single array, thousands of single cells are dividing and developing into viable monoclonal colonies. Now, thousands of validated clones sounds amazing, but how do you navigate through all of those clones to find the ones you really want? Enter our GPS, the brains of the technology, the CellRaft cytometry software. Using bright field images acquired on our instrument, the CellRaft Air, the software allows the user to define the exact criteria for the cells or colonies that fit their experimental needs, from size to morphology to fluorescent marker expression. After you've identified your clones of interest, it's time to retrieve them. Using the automated CellRaft Air, each CellRaft can be individually isolated from the array to a 96-well collection plate with its precious cell cargo still attached, without the need for fluidics, enzymatic dissociation, or manually scraping that could harm your cells. It's not magic, it's magnets. On the Air, colonies are isolated by gently dislodging the CellRaft from the bottom of the array. Because there are nanomagnetic particles integrated directly into the raft, as the raft is released, a magnetic wand retrieves it from the top. The raft, with cells still attached, is then carefully and safely deposited into a collection plate. Your 96-well plate is automatically filled, hands-free, with the desired cells or colonies for downstream experiments. And most importantly, that 96-well plate is filled with already established, viable colonies or organoids, not single cells, putting you miles ahead on the road to success. Want to learn more about how this technology can put your science in the fast lane? Visit our website at www.cellmicrosystems.com, request a demo, and get in the driver's seat in your own lab.